Hello. My talk today is about integrated in silico and experimental assessment of the disease-relevant or poor coherent 19 missense variants. We all know that mutation in this gene causes female epilepsy, which for this talk I will refer to as PCH19 GCE. The molecular mechanism of this disorder remains largely unknown. We recently found that PCH19 can be present in the cell nucleus of MCF7 cell line, which is a breast cancer cell line. Here is just a picture of what an MCF7 cell lines look like under the fluorescent microscope. There's a cytoplasm, and that's the nucleus. We found that PCH19 can be present in the nucleus of this cell line with NO and estrogen receptor alpha. And together, they co-regulate gene expression. We also developed an in vitro assay system which allow us to measure the effect of PCH19 on gene transcription. This is a estrogen response element reporter assay system. Just let me quickly demonstrate to you how this assay works. Under a normal physiological condition, there's NO and estrogen present in the cell lines, and if we transfect in the reporter assay, this leads to basal transcription activity. And if we treat the cells with estradiol, which is the ligand that interact with estrogen receptor alpha to activate it, we have increased transcriptional activity. And when we transfect in the wild-type PCH19, this also leads to increased estrogen receptor alpha transcriptional activity. And this activity is further increased when we treat the cell with estradiol. However, if we put in one of the disease-causing PCH19 variant, this leads to a significant reduction in transcriptional activity, which cannot be rescued in the presence of estradiol. To measure the effect of PCH19 on the physiological effect of PCH19 on transcription, we also look at some of the endogenous target of estrogen receptor alpha. What we do is we transfect in, again, either wild type or one of the PCH19 disease-causing variants, and measure the level of the expression level of these endogenous targets. And here you can see, if we transfect in wild-type PCH19, this leads to an increase in these endogenous targets. However, when we put in one of the PCH19 disease-causing variants, the level of these endogenous targets is significantly reduced. We also have been able to demonstrate that this endogenous target is disrupted in the skin fibroblasts of the patients. Here you can see, this is the level of these endogenous targets in the control individuals. And then their levels are affected when we look at the skin fibroblasts of the patients. Together, these results suggest to us that what we see in our in vitro assay system is biologically relevant to what we see in the skin fibroblasts of the patients, which give us confidence to use this assay to assess more PCH19 variants. This is a busy slide, but this is just to show you what a PCH19 protein looks like. We have the extracellular domain, which consists of 6 coherent repeats, a transmembrane domain, and also a cytoplasmic region. Here are the location of 146 unique missense variants that has been implicated in PCH19 GCE so far. You can see that the missense variants make up the most number. However, when reviewing the literature, we found variants of unknown significance and also variants that are found common in the human population are also reported to be as causing PCH19 GCE. Thus, it remains a huge challenge to differentiate between what is likely benign and what is likely pathogenic, which makes it even more difficult for us to assess the variants of unknown significance. Also, I just want to tell you like a small example of what happened. What we experienced is that a little girl that came to us, she was diagnosed with PCH19 GCE because she carried a PCH19 variant. However, a few years later, we found that in addition to the PCH19 variant, she also had mutation in 3 other genes, and 2 of them are likely to be pathogenic. We have also included this variant in our assessment, and we found that this PCH19 variant is like benign. So, therefore, we believe that correct assessment of this variant is very important because it can improve correct diagnosis, which can help us guide treatment for the patients. So this leads us to our study, is to assess 31 PCH19 missense variants. But first, we group these variants into 3 different groups. So first, the PCH19 disease-causing variants. So these variants include both unpublished as well as published variants. The variants of unknown significance. So these variants come from the EPI4K as well as the Phenome Genome Project with allowed frequency of less than 1 in 10,000 according to the normal database. So this is a database which contains common variants found in the human population. Also the high frequency variants. So these variants have allowed frequency of greater than 1 in 10,000 according to the normal database. And we use in silico predictions as well as protein structure modeling and in silico functional analysis to assess these variants. So here is the summary of our in silico assessment of the PCH19 variants. So this graph is plotted using CADFRED and PolyFriend. So these are 2 computational tools that's taking into account of different factors with higher scores indicating damaging. So you can see the majority of our PCH19 causing disease variants are located in the top right corner, suggesting that they are damaging, while the majority of the high frequency variants located in this bottom left, suggesting that they are likely benign. And the variants of unknown significant clustered throughout the graph, which is what we expected. We also use protein structural modeling to assess this variant. So this work is done together with collaboration with our collaborator in Adelaide, Australia. So to be able to do this, we built a PCH19 homology structure based on 2 published crystal structures, the zebrafish PCH19 EC1 to EC4, and the mouse PCH17 EC5. So from this crystal structure, so this is the first PCH19 homology structure, which includes EC1 to EC5, that include both calcium binding sites and glycosylation sites. So glycosylation is a process that adds sugar molecules to protein to increase the stability and the functionality of the protein. So from this homology structure, each structure was built for each PCH19 variant. So each of these variants were subject to energy minimization to determine whether if the change affect protein instability. So here is an example where the PCH19 variant lead to protein instability. Also, amino acid that had a proline change add a kink to the amino acid chain of the protein, which also disrupt the secondary as well as tertiary structure of the protein, and that's also lead to protein instability. So here is an example of PCH19 variant that has a proline change that leads to protein instability. Also, each of these variants was visually inspected to see whether if the amino acid change is conserved across multi-species, to see whether if it's lies within a glycosylation site, So here are some examples of PCH19 variants that affect glycosylation sites, and to see whether if the amino acid change affect calcium binding sites. And here are some examples of PCH19 variants that affect calcium binding sites, and to see whether if the amino acid affect hydrogen bonding interactions. So here are a summary of our protein structural modeling. So for the PCH19 disease-causing variants, we had 8 out of 10 of these variants affect the protein structure. For the variants of unknown significance, 6 out of 13 of these variants affect the protein structure. And for the high-frequency variants, the majority of these variants located in the C-terminal region of the protein where we don't have a protein model available. But for those that we have the protein structure available, 1 out of 3 of these variants appear to affect the protein structure. We also use our system to assess this variant. So here I just want to demonstrate to you how each of the variants from different groups behave in our functional assay. So again, when we transfect in wild type PCH19, we have increased transcriptional activity, and this activity can further be enhanced when we treat the cell with estradiol. And when we transfect in one of the high-frequency variants, these variants behave like a wild-type PCH19, which also increase estrogen receptor alpha transcriptional activity either in the presence or absence of estradiol. And when we transfect in one of the PCH19 disease-causing variants, this leads to a significant reduction in estrogen receptor alpha transcriptional activity, which cannot be rescued even with estradiol. And the variants of unknown significance have mixed results in our functional assay. So here are the summary of our functional analysis prediction. So for the PCH19 disease-causing variants, we have 9 out of 10 of these variants appear to affect the protein function. And for the variants of unknown significance, we have 3 out of 10 of these variants also appear to affect the protein function. However, for the high-frequency variants, only 1 out of 4 of these variants appear to affect the protein function. We also look at the physiological effect of these different PCH19 variants by looking at the protein level of 2 of the estrogen receptor alpha endogenous targets. So here we look at oxytocin receptor. Oxytocin receptor play a very important role in reproduction, and also it has also a role in behavioral disorder. And AKR1C3 is the enzyme, if you remember, that's responsible for converting 5-alpha-DHP to allopurinol. So here you can see, so we look at the protein level of these two endogenous targets by Western blotting. So here's the result. So when we transfect in the wild type, we see that there's an increase in the protein level of these 2 endogenous targets. If we transfect in one of the high-frequency variants, it also behaves like a wild type, and that also increased the protein level of these 2 endogenous targets. And if we transfect in 2 of the disease-causing variants, this leads to a reduction in these 2 endogenous targets. The variants of unknown significance show mixed results, but in this case, it's behaved like a wild type PCH19. We also look at these 2 endogenous targets in the skin fibroblasts of this patient. So first we look at oxytocin receptor, and here you can see oxytocin receptor mRNA as well as protein are significantly higher in the affected patient as compared to control. We also look at AKR1C3. You can see both the mRNA and also the protein level are significantly lower in the affected individual as well as compared to the control. So as mentioned earlier, these are two of the endogenous target of estrogen receptor alpha. So we have seen that both of these endogenous targets are disrupted in our in vitro SI system as well as in the patients. So which suggesting to us that this is what we see in our in vitro assay system is biologically relevant what we see in the patients. So in summary, we have tested 31 PCH19 variants, and 21 of them we could test across all 3 pathogenicity assessment tools. So 18 out of these 21 variants received the same prediction outcome from all 3 prediction methods. Almost all PCH19 disease-causing variants predicted to be pathogenic, affect the protein structure as well as function, with a few exceptions which are listed below here. And for prediction of PCH19 disease-causing variants and the high-frequency variants, although the numbers are still very small, but we believe that our in vitro analysis was the best performed method. So a combination of these prediction tools was able to distinguish between PCH19 disease-causing variants versus the tolerant variants with reasonable accuracy. So we hope to combine these in silico and in vitro tests to test new reported PCH19 variants. So we hope that we can use them to improve diagnosis for PCH19 patients, guide clinical trials and treatment for these patients. Thank you very much. And finally, I would like to thank my boss for letting me to carry out this work, and also all the people that's involved in the work. Thank you very much for listening.
Thank you very much. I'm sorry, I had no chance to hear all the presentation, but only the first part. Sorry about that. Do you have questions from the audience? There's one there. Thank you.
Thank you very much for the great talk. What is a part of the protein which is translated to the nucleus and modify the gene expression? This is the C-terminal, the N-terminal part?
So in this cell line, we can find that the full-length PCH19 can also localize in the nucleus.
But physiologically, we know which part is translated?
We found that if you transfect in just the C-terminal region of the protein, because that region as predicted, it has 3 nuclear localization signals in the C-terminal region of the protein. So if you transfect that in, then that goes straight to the nucleus. Whereas if you transfect in the wild type, the full length, part of it goes to the nucleus, but also part of it are present throughout the cells as well.
So we can test with your assay all the mutations, whatever the localization on the protein?
Yes.
Okay.
Okay, thank you so much. We need to move to the next session. That is the 4th session, stem cell models, and